Imagine, for a second, if you could see the invisible. I mean, actually watch, in real time, as tiny proteins build the machinery of life inside a living cell. For decades, that was pure science fiction. But then, scientists found a key, a kind of magic lantern, and they found it in the most unlikely place you can think of, a tiny, glowing jellyfish. So take a look at this. On the left, that's the jellyfish Aquaria victoria, glowing all on its own. And on the right, that's a labworm, C. elegans, shining with that exact same light. Now, this isn't just some cool party trick. This was a complete game changer. The ability to take the glow gene from one animal and pop it into another, well, that gave science an incredible new superpower. And that superpower was aimed squarely at a huge problem. How do you possibly track one specific molecule or a single cell in a living thing without messing everything up? It's kind of like trying to follow just one person in a massive, cheering crowd, but the lights are out. Scientists needed a beacon, a tag, some kind of biological lantern they could just attach to whatever they wanted to study and watch it move. The story of how we got this lantern is just a fantastic tale of scientific curiosity, a little bit of luck, and some amazing collaboration. It's a journey that literally started in the ocean and ended with a Nobel Prize. You can think of it like a relay race between these incredible scientists. It all starts back in 1962 when Osamu Shimomura first pulls this green fluorescent protein, or GFP, out of a jellyfish. For years, it was just a scientific curiosity. Then boom, 1988, Martin Chelfie has this amazing aha moment. He realizes this could be the perfect tag for the see-through worms he studies. By 94, he proves it works. And then Roger Tien grabs the baton and just supercharges the whole thing, making it brighter and creating a whole rainbow of colors. What's so amazing is that this all kicked off with pure fundamental research. Shimomura wasn't trying to invent a tool for cell biologists. He was just curious. He just wanted to know why this jellyfish glows. And his dedication was unbelievable. Over 19 years, his team collected an estimated 850,000 jellyfish, just to get enough of this protein to study it. I mean, wow.
Okay, so we've got this amazing glowing protein, but how do you actually get it inside a specific cell and make it light up when you want it to? It sounds like something out of a sci-fi movie, but it's now a basic technique in labs all over the world.
So let's break it down into a simple 3-step process. First, scientists get the exact genetic code for GFP. Think of it like finding the instruction manual that says how to glow. Step 2, they stick that instruction manual into a delivery vehicle, usually this little ring of DNA called a plasmid. And then, step 3, they get that vehicle into the cells they want to study. The cell's own machinery reads the manual and just starts building the glowing protein. Simple as that.
Of course, the reality in the lab is a little more complicated than just 3 easy steps. This flowchart here gives you a much better sense of all the work involved. It's a super careful, multi-stage process, but honestly, that 3-step overview we just went through, that's the core of the magic right there. For years, GFP was pretty much seen as the perfect tool, a harmless little light bulb you could stick anywhere. But as more and more researchers started using it, a darker side began to show up, a kind of shadow cast by the magic lantern itself. And this led to a really critical, kind of uncomfortable question that scientists had to ask themselves. Is this incredible tool, which let us see so much, actually changing the very things we're trying to watch? And it turns out there were 2 pretty big problems. First is something called cytotoxicity. The GFP protein itself can actually be toxic. For some cells, it acts like a slow-acting poison, creating damaging molecules and even telling the cell to self-destruct. The second problem is immunogenicity. Your immune system is built to attack anything foreign, right? Well, a jellyfish protein is about as foreign as it gets, so the immune system would hunt down and destroy the very cells that researchers were trying to study.
And these weren't just theories, you know? The evidence started piling up from all sorts of experiments, showing that this glowing tag wasn't always just a quiet observer. The findings, and you can see a summary here, were really troubling. On the immune side, you had T cells, the soldiers of the immune system, getting activated to kill the GFP cells. And on the toxic side, the problems range from triggering cell suicide, what's called apoptosis, to causing direct damage to heart and muscle cells. This created a huge problem. I mean, if your glowing cells vanish, how do you know if it's because of the biology you're studying or if it's because the tag itself made them die? And here is just one really stark number to drive the point home. In a study with rat liver cells, the ones that were given GFP, they produced 50 times fewer stable colonies than cells that were given its less toxic cousins, the cyan and yellow proteins. 50 times, that is a massive, massive difference, and it just shows how much the tool could mess with the experiment. But this is what science does best, right? It finds a problem, it studies the heck out of it, and then it engineers a solution. The story of GFP doesn't just stop with its flaws. It continues with all the clever ways researchers figured out how to get around them. So, how'd they fight back? Well, they developed a whole bag of tricks. They started choosing specific strains of mice that were less likely to freak out about the GFP. They figured out that using animals born with the gene was way safer. They even used immunosuppressant drugs. And most importantly, they went back to the protein itself, found ways to use less of it, and as we just mentioned, they developed much better, less toxic versions. And the ultimate result was, well, it was stunning. That initial problem with GFP's toxicity actually pushed the entire field forward. It led to the creation of this entire rainbow of fluorescent proteins. Scientists can now literally paint cells with a whole palette of colors, like in this incredible rainbow image. It lets them track tons of different things all at once and do it more safely and more beautifully than ever before. You know, the story of GFP is just such a powerful reminder that pretty much every revolutionary tool in science, from the first microscope to CRISPR, comes with its own hidden rules and limitations. It really makes you wonder, doesn't it? What are the subtle, unseen flaws in the tools we rely on every single day? And what amazing new science will happen when we finally find them?